So y'all can see my slides. In this presentation, I'm not going to talk about HO1, but really some advances in the field of nephrology. I picked 10, so these are not perfect. These are my choices that I think have really made a fundamental difference in kidney disease. So these are my disclosures. None of these are relevant to today's presentation. So why is it really important? If you look at the facts, 850 million people worldwide have kidney disease, 37 million Americans. So that's about almost 14% of the US population that have kidney diseases. And if you look at the impact, it's the 8th leading cause of death. 130 billion is spent for managing kidney disease. Every year, 100,000 people start dialysis, and half of them will die within 5 years. 9 in 10 people don't know that they have kidney disease. And 1% of the Medicare population here has kidney failure, but 7% of the budget is required to treat kidney disease. So you can just see how much the burden is, that how much we need new therapies, new discoveries in the field of nephrology. So these are the top 10 advances that I will go over. I'll provide some basic knowledge and highlight how that basic knowledge led to that clinical discovery in some cases. And in a lot of these instances, there was a trainee that was involved, whether a medical student, a resident, a fellow, a postdoctoral researcher, as the first author on the work that really led to this seminal discovery. So let's talk about SFLT1 in preeclampsia. So what is preeclampsia? It's a syndrome affecting about 5% of pregnancies. If you have preeclampsia, very high mortality for both the mom and the baby. Before this paper was published, nobody knew what the pathogenesis was. They thought it was endothelial injury, vasospasm, platelet activation, prostacyclin, thromboxane imbalance, nitric oxide. But they knew one thing: the placenta was critical. If you took the placenta out, the mother would become normal, the baby would also be normal. So that clearly everybody knew that the placenta was key. This is just a figure to show you that the placenta plays a central role, whether it is genetic, environmental, or other factors, that leads to vascular endothelial dysfunction and high blood pressure, kidney failure, seizures, liver dysfunction, and so on. And that's the syndrome of preeclampsia. So this work that was published in the JCI in 2003, Sharon Maynard was a fellow at that time at the Beth Israel Deaconess Hospital. They found this protein that was critical in the pathogenesis of preeclampsia. How did they find this out? So they took patients with preeclampsia, they took normal, healthy, normotensive pregnant women, and they took the placentas, they ground it up and did a microarray analysis. They also used an animal model of preeclampsia. They did gene expression profiling of the placentas for the women with and without preeclampsia, and this is what they found. So you can see here, N1 refers to normal, P1 refers to preeclampsia, and these are 3 patients, P1, P2, P3. You can see in only the preeclampsia, the levels of this protein, SFLT1, were significantly higher. And you can also measure it in the blood. Very high levels in the blood of severe preeclampsia, even mild preeclampsia, the levels were significantly higher. They injected SFLT1 using an adenovirus construct into an animal model of pregnancy. And what they found was the normal control injected FC, part of the antibody, didn't really have any issues. But the protein SFLT1 injection resulted in the classical histological changes of preeclampsia, namely glomerular endotheliosis, high blood pressure, and proteinuria as well. They took this to the clinic. They published this New England Journal paper showing that these factors would actually predict onset of preeclampsia. So you can see here in the red is women who later developed preeclampsia. And in the black here, women that had clinical preeclampsia. But even before you get preeclampsia, levels of this protein go up. So you can predict that, hey, you may develop preeclampsia. So we may need to handle the pregnancy, terminate the pregnancy earlier or deliver the child, if it is viable. More recently, they have now doing different manipulations. Can you modulate placental SFLT1? Is SFLT1 bad for you coming from the placenta? Can you block that using RNAi? There are certain drugs like statins and others that block SFLT1. So you see how that basic discovery in the pathogenesis of preeclampsia is now leading to therapies, not only as a biomarker that SFLT1 may be important, but in terms of treatment for preeclampsia that can help both mother and the baby to not have this complication from preeclampsia. Also note the timeline. The first discovery was in 2003. It has taken 20 years to get to where we are today. So science takes a long time, to translate into the clinic. We've seen some very rapid translations, obviously with COVID, with the vaccine and so on. But again, the technology keeps changing, every year, every decade, and things are happening much quicker. So let's talk about this PLA2R in membranous nephropathy. So membranous nephropathy is a very common cause of nephrotic syndrome in adults. It's thought to be antibody-mediated autoimmune disease. And if you do histology, you have immune deposits within the kidney, but nobody knew what the antigen was that was creating this antibody response. So in this work, again, another fellow, Lawrence Beck, found that this M-type phospholipase A2 receptor was the target antigen for membranous nephropathy. They took patients with membranous nephropathy, they took their kidney biopsies and glomerular extracts and did Western blots and found that this protein, 185 kilodalton protein, was increased in membranous nephropathy, denoted here by MN1, 2, 3, 4, and 5, and other causes of nephrotic syndrome like FSGS or diabetic nephropathy did not have this protein increase. They also were able to find serum levels of this protein to be increased with membranous glomerular nephropathy. And they also found it in the kidney. Here's a control IgG stain in green, and in the red is this PLA2R staining in the glomerulus all across these cells. And in lupus, a secondary cause of membranous nephropathy, you don't get any staining. So this is very specific to idiopathic or primary membranous nephropathy. More importantly, this correlates with activity of the disease. So higher levels of this protein, PLA2R, correlates with immunological activity and clinical disease. And if the levels go down, it actually signifies remission. So really an important biomarker and also a pathogenic factor. So 80% of patients with primary membranous, this levels are increased. It's very specific. You can monitor disease activity. You can predict a remission or a relapse. And if you get kidney failure from membranous, and if you have a transplant, if this antibodies appear after transplant, it's high risk for recurrence of your disease within the kidney. So you can see, again, an amazing discovery that has led to clinical management of patients with membranous nephropathy. We'll talk about ApoL1 in kidney disease in African Americans. So in this work, another trainee in the lab of Martin Pollack, Dr. Giulio Genovese, he found this association of trypanolytic ApoL1 in kidney disease in African Americans. So it is known that in African Americans or people who lived in Africa, they're very susceptible to this disease called African sleeping sickness. And the disease that causes it is this bug, Trypanosoma brucei, that is transmitted through an insect. In Africa, over millions of years, people have actually developed mutations in this gene called ApoL1 that actually makes them resistant to sleeping sickness. But as Africans migrated to the United States, these variants, G1 and G2 variants, actually increase risk for African Americans. So if you have an African American with the same kidney disease that another Caucasian American has, they have almost 10 times higher chance of losing kidney function, whether it's hypertension, diabetes, or any other kidney disease. So clearly there's a very high risk for African Americans with kidney disease. So what they found was that this G1 and G2 variants are actually responsible for this high incidence of African Americans having kidney disease. Further work from Catalin Shustak and colleagues found that if you overexpress these G1 and G2 variants in animal models, you can reproduce the human disease. So this is like fulfilling Koch's postulates. You find something, You go back to patients or you go back to animals and prove the point. So here's data. The top 3 panels are normal controls. So G0, G1, and G2. So you can see G0, the kidneys look completely normal. But if you have one variant or 2 variants, G1 and G2, in the high magnification in the lower panels, you can see classical changes of glomerulosclerosis, a progressive kidney disease that is very commonly seen in African Americans. This marker is really important in kidney transplantation. There's a national NIH institute-funded study looking at these risk variants in donors or recipients after kidney transplantation. And more recently, a company, Vertex, has devised an inhibitor. It's a small molecule, orally available ApoL1 inhibitor, VX147, that has now been shown to be very effective in these patients that have this gene mutation for focal segmental glomerulosclerosis. And this was a paper published last year in the New England Journal. The drug is called inaxaplin. And for proteinuric diseases in people that have the 2 variants of ApoL1, significantly reduces proteinuria and the risk of developing kidney failure. Only earlier this month, they actually have moved it into a phase 3 study as a top-line prospect for this company, Vertex, which is well known for developing new therapies for diseases like cystic fibrosis and so on. So hopefully this larger study that was just reported earlier this month will take this drug to the next level for late-stage testing in a large clinical trial for these patients with ApoL1 gene mutations. Let's talk about artificial intelligence. We hear a lot about artificial intelligence in various fields, even outside of medicine. But nephrology is making a significant headway. In fact, this has been going on for at least 5 or 6 years now. And I'll show you one example. There are several examples, but one example of how AI is being used now for clinical care. So this was work where using machine learning, they were able to subphenotype sepsis-associated acute kidney injury. So a lot of patients in the intensive care unit will develop kidney failure. And most of that is from sepsis. That's the commonest cause that I get consulted to see a patient in our hospital is for sepsis-associated AKI. So these group of people, they looked at 4,000 patients with sepsis-associated AKI. They just looked at the electronic health record, the EHR. Whatever data was built into the EHR, nothing special, and they applied deep learning tools, the vital signs, the labs, the comorbidities, and they were able to subphenotype these patients into 1, 2, and 3. And you can see on this graph, very distinctly categorized 3 subgroups in the blue, green, and the orange. And why is this important? Because it matters whether you need dialysis or if you die. So subphenotype 1, very low need for dialysis, and the mortality was about 20%. Subphenotype 2, much higher need for dialysis and mortality almost 35%. But if you were subphenotype 3, very high incidence of need for dialysis, and your chances of mortality was almost 50%. So if you have a treatment intervention, and if you give it to all the 3 subphenotypes without categorizing it, you may not get a benefit. So there's a lot of noise in the system, so the treatment doesn't really pan out. But if you subphenotype and then implement the treatment, then you have a chance of using that treatment, making it further into clinical care. So a lot of now clinical trials are being done by using these AI tools to subphenotype different kidney diseases and other diseases as well, in cancer and so on, to target the right treatment for the right patient to see if that will work or will not work. Let's talk about NAD metabolites. So the mitochondria are very critical in organ function. So after the heart, the kidney has the highest number of mitochondria within the proximal tubules. And this protein, PGC1 alpha, is a master regulator of mitochondrial function. And a protein that is important is NAD. And NAD is part of the oxidative metabolism. And in this work, several years ago, they showed that PGC1 alpha really drove NAD in terms of mitochondrial biogenesis, and in an animal model, it was shown to protect against acute kidney injury. The same group took this to the clinic. So they were able to use NAD supplementation and showed that that had a profound impact in reducing acute kidney injury even in humans. So what they did, you can actually buy NAD from a store. You don't even need a prescription. It is niacinamide. 500 milligram, they gave placebo or nicotinamide 1 gram or 3 grams. 1 day before cardiac surgery, day 0, and on day 1. And they followed up these patients. And you can see the data here. It's a pilot study. In the red is the placebo-treated group. Following cardiac surgery, you have a high chance of getting kidney failure. They had kidney failure, but the ones that got either the 1 gram or the 3 grams had significant protection compared to the placebo group. So this group is now looking at larger clinical trials to see if this really, really pans out. Again, fundamental science that led to this treatment intervention for these patients. Let's talk about circadian rhythm and cardiorenal function. More than 15 years ago, Michelle Gums, and she was a graduate student at that time in nephrology, showed that this protein, PER1 or period 1, regulates expression of the sodium channel in mice. And now there's a lot of work going on with how can you regulate this protein, PER1. There are other proteins as well, BMAL and so on, that they actually affect kidney disease. That's why you have to time your intervention of giving medication. Take it at 8 a.m. or 8 p.m. There's a reason behind it, because it depends on your circadian clock and how that regulates different proteins. And we know that the Nobel Prize was awarded in 2017 to the discovery of the circadian rhythm proteins by Dr. Hall, Dr. Rajbash, and Dr. Young. So remarkable discoveries, again, going to decades of research into the circadian rhythm. And there's going to be a lot more coming out of this discovery, not only in nephrology, but in other fields as well. We'll talk a little bit about SGLT2 inhibition for kidney disease. And I'm sure in clinic, you all are using, these drugs called Flozins. So how did these work? Again, pioneering work going back to the 70s and 80s by investigators at UC San Francisco showed that if you block this transporter in the proximal tubule of the kidney, sodium glucose transporter type 2, which is responsible for 90% of the glucose that is filtered back, into the system through these transporters, that this inhibitor blocks this and essentially can have profound effects in managing diabetes. So they did multiple clinical trials, and one of the drugs, canagliflozin, they're traditionally referred to as Flozins. This paper was the Credence trial, showed very nicely that this drug actually preserves, urine albumin to creatinine excretion is significantly reduced in the canagliflozin shown here in the blue line. And also estimated GFR is preserved in the patients that got this drug, canagliflozin. You can see large numbers, over 4,000 patients, an international, clinical trial with multiple centers. And there have been other drugs related to the same family. This paper published in January of 2023, empagliflozin in the EMPA Kidney Collaborative Group study showed very similar findings, reducing proteinuria and preserving kidney function. And this was in addition to standard of care with ACE inhibitors and angiotensin receptor blockers, that these drugs have great protective effects, not only in the kidney, actually in the heart and other organ systems as well. It is now standard of care to use these drugs in our patients. Along the same lines, endothelin-1 antagonists have also shown to be very effective in preventing progression of kidney disease in type 2 diabetes. This was the first paper using atrasentan and renal events in patients with type 2 diabetes, or the otherwise called the SONAR study, a double-blind clinical trial published in the Lancet. And more recently, just a few months ago, another related agent, sparsentan, versus arbosentan, irbesartan, which is an angiotensin receptor blocker in focal segmental glomerulosclerosis, that it has protective effects even better than actually atrasentan. So again, newer drugs being available in our armamentarium for our patients with kidney disease. This is very hot off the press from just a few weeks ago, kidney xenotransplantation. There are over 100,000 people waiting for a kidney transplant in the United States. I don't know what the data is for Poland and Europe in general. We don't have enough organs to be transplanted in patients. So a lot of work over 25, 30 years is being, can we use pigs or other animals to generate organs? So it was really difficult until the advent of CRISPR-Cas became available. So using CRISPR-Cas, several groups have been able to develop transgenic pigs that allow you to at least test the model that these pig kidneys can be used in a human being. So before doing studies in living human beings, we published this paper in 2022 using a human decedent model, which essentially somebody has died, their organs cannot be used for transplant, but they're willing to give their body up for research. So we can keep their heart and lungs beating, they're brain dead, so we can take their kidneys out and show by a kidney transplant that that kidney does not get rejected immediately by hyperacute rejection and can stay for a few days, for a few weeks, for a few months. And this was shown here, where we took a brain dead person, we took the 2 kidneys out, we did the kidney transplant using this 10 gene edited pig, and Joseph would like this. One of the genes that is overexpressed in this pig is the HO1 gene, because of its anti-inflammatory, immune antioxidant properties that was overexpressed in this gene to preserve organ function. And the results have been really impressive using this 10 gene edited pig. We've done about 5 of these since this first transplant in April 22. Several other institutions have done it. They've done 2 heart transplants in living people. Some of those hearts have lived for 2-3 months after transplant. This work from Mass General, from a company eGenesis, developed a newer transgenic pig that has 69 gene edits. The concern is some of the viruses in the pigs can cause zoonosis and viral infections in humans. So there was concern. So they made an even better pig model of 69 gene edited pigs, and only so this was 2 other studies using the pig to human kidney xenotransplant in the decedent model. But this is again March 22nd, last month. The 69 gene edited pig was transplanted into a living person through compassionate use. It was a 62-year-old patient. The patient had a minor episode of rejection, but has gone home and doing quite well now, almost 3 weeks after transplantation. So a lot of excitement in the field. Our center is going to the FDA to see if they would approve us to do a clinical trial for using our 10 gene edited pig model that was developed by United Therapeutics. So a lot of excitement, in this regard, because this will essentially eliminate the need of whole scale dialysis for our patient population. So a huge advance. And again, technologies like CRISPR-Cas enable this to really happen. And finally, a lot of advances in single cell omics, precision medicine, and kidney nephrology is not far behind. The NIH felt this was really important, and 5 years ago, they funded a multi-center project called Kidney Precision Medicine Project, or KPMP. The focus is on human kidney disease. How can we devise individualized treatments? How can we improve the young investigator pipeline for people to pursue, kidney disease research? And why did they do this? We want to develop new therapies for acute kidney injury and chronic kidney disease. A lot of animal models have been very successful for, showing that therapies work, but when they tried to do it in humans, it never worked because we didn't have human kidney tissue. So the goal was to create a human kidney atlas so we can better understand what are the pathogenesis of human acute kidney injury, of human chronic kidney disease. And there's a lot of heterogeneity in humans. So can we really understand that better and then use it for therapy? So some unique aspects, it includes patients, clinicians, scientists, ethicists. Patients are equitable partners. They have an active voice in every part of the research enterprise. So really will address a big public health problem. I told you 850 million people worldwide have kidney disease. To really use this kidney atlas focused on human, kidney pathology. So they've done biopsies. I just accessed this website this weekend. 242 patients with chronic kidney disease. They've done kidney biopsies, 66 with acute kidney injury, 18 with diabetic kidney disease that they didn't really have a clear cause, and 125 control biopsies that have been done and that have collected tissue. And they published a beautiful paper. It's over 100 pages with, over 200 pages of supplemental material. They created this atlas of not only a healthy kidney, but also injured kidney, the human kidney, what happens to the different cell compartments. The kidney is complicated because it is not just one cell type. There are over 100 different cell types within the kidney, and each cell type has a specific function. There are specific proteins, there are specific transporters that are affected in a given disease. So unlike a lot of other organ systems, the kidney is very intricate, very complicated organ system that why it's been difficult to target different drugs to the right cell type. So hopefully this kidney atlas will really enable that for us to be able to do it. So they're taking the clinical parameters, they're looking at the demographics, they have a kidney biopsy, they're biospecimens, they're doing transcriptomics, pathomics, proteomics, metabolomics, epigenomics, and they're integrating all that data in these people, putting it together, making it all available freely, open access. So if you go to the website, kpmp.org, all this data is available for free. Hopefully this kidney atlas will allow us to develop targeted therapies, platforms for continuous discoveries, and really a great resource for patients and other key stakeholders. Like the consent form for these patients to develop kidney biopsies was developed by the patients. So really, it's not the scientists or the doctors telling patients, do this and don't do this. The patients are at the center of the table making decisions for their care. So they're using a lot of single cell technologies, trying to understand what these different 100 cell types, are doing within the kidney. They're using spatial technologies, pathomics, clinical parameters, and really putting all this together so they can map cell-specific signatures and then use that to determine, what should be used, what target should be identified for treatment. So the goal is to really understand and treat human kidney disease ethically and safely, obtain these kidney biopsies, create this kidney tissue atlas. And so we'll find these subgroups. I showed you that AI worked with a subphenotype patients just based on clinical data. But now we can subphenotype based on all these biological data, identify critical cells, pathways, and targets for novel treatments, develop individualized treatments, improve our scientific knowledge base, and more importantly, help our young investigators continue to pursue science in the field. So again, I talked to you about how this is really critical. So hopefully I've convinced you that there's been great progress in the last decade in our field in nephrology. A lot of new diagnostics, precision medicine and mechanism-based, therapeutic approaches. So hopefully a lot to look forward, to the future of nephrology. I'll stop here. And this is just the skyline of our city, Birmingham. Thank you for your attention.